Welcome to Click for IB Biology. We're studying genetics and now we're looking at dihybrid genetic crosses using unlinked genes. There's a blank version of this presentation. A PDF is down in the description. You could download that and annotate with your own notes. There's a quiz down there of 10 questions with answers, and you can use that to check your understanding of the content of this part of the course. So this particular dihybrid cross that we're going to do is taken from Mendel's work and will serve as a model for dihybrid questions. And in this process, the first thing that we do is to write down the phenotype of the parents, and we designate the parents P, so we could actually call this one parent 1, and this one will be called parent 2. Now you can see that there's two characteristics, two traits, because this is a dihybrid. So remember that dihybrid involves two characteristics or two traits, and the traits to be dealt with are the color of the pod and the color of the seed. Now in this particular cross, what we know is that the green pod is dominant to the yellow pod. That should be a G, little g like that. And that the yellow seed is dominant to the green seed. So easy to get confused with the use of these letters. So very important that you take your time and work through this systematically. In my particular example, I'm going to begin with two parents who are homozygous double dominant, homozygous double recessive. And so my genotypes can be written down as big G, big Y, little g, little y. And we know that those letters represent the base sequences to be found on the two genes in the two separate homologous pairs. Now then, when we go through meiosis, this is a fairly straightforward one, that because we separate the homologous pairs, so we're going to have big G, and we separate this homologous pair, so we're going to have big Y. The same can be done on the left, with small g's separate, the small y's separate. And even though we observe the law of independent assortment, this isn't going to make any difference to our gametes because they are homozygous at the two gene loci. And so all of the gametes will be big G, little y from this parent, and all of the gametes will be little g, little y from this parent. And so when that fertilization occurs, we would end up with big G, big Y, little y. So that's two new homologous pairs. This is the fertilization. And so we can then write down the next section, which is our offspring genotype, which is big G, big Y, little y. And we note that all of the gametes, all of the offspring would be of that genotype. And then let's turn that to finish off with into the actual phenotypes, which would make the offspring all green pods and all yellow seeds. So we had P1 times P2, homozygous double dominant, homozygous double recessive, and we end up with the F1 generation, in which they are all heterozygous at two gene loci, making them big G, big Y, little y. So that's our heterozygous F1 population. So let's move on from this point. And let's take the two heterozygotes, and now what we're going to perform is known as an F1 cross. We'll take one individual, one plant, so there would be one pea plant, another pea plant. These are both heterozygotes for the two genes that we are heterozygote. For the two genes, they are both heterozygotes. And so if we just look at their phenotype to start with, so let's write down, these are the parents' phenotype. They would have green pods and yellow seeds, green pods and yellow seeds. However, we know that their genotype is big G, big Y, little y, big G, big Y, little y. So now what we have to do is to consider the possible outcome of meiosis. And of course, if I just work over here, both parents are going to be the same. So let's just do one of them using the method I showed you earlier. So this means that we must separate the homologous pairs. So we would end up with big G with big Y. That's one gamete. Big G with the little y. That's the second gamete. The little g could end up with the big Y, and because of the laws of independent assortment, we would also have little g and little y. So 4 gametes to line up on this side here, like so. And of course, that's going to be equally true on the other side. So let's look at the completed table for this process. Now, these are the 4 gametes on this side. These are the 4 gametes on this side for this F1 cross, which is an F1 crossed with an F1. Crossed means to cross-pollinate, to reproduce them together. And so if big G, big Y is fertilized by big G, big Y, then this is the genotype we would end up with. And so now I've filled in the phenotypes immediately, showing green pod and yellow seed. Now you might like to just pause the video and work your way slowly through these combinations of gametes and the resultant fertilizations. The fertilizations are the 16 boxes here in the Punnett grid. These are the 16 possibilities, and what would be interesting for you to do is to score the frequency of the phenotypes. So perhaps you might like to just pause the video now and go through these fertilizations by yourself. Okay, so if we then write down our offspring genotypes, 1, 2, 3, 4, 5, 6, 7, 8, 9 different genotypes are possible. But when we look at the phenotypes, we discover that 9 of the 16, so 9 out of 16 possible genotypes would give us green and yellow. So that's double dominant. 1 out of 16 would give us the double recessive yellow and green. And then here we have 3/16 has the dominant green pod, but the recessive P color. And then in the other 3/16, there would be a yellow pod and a yellow seed, like so. So that's recessive here, but dominant here. And so this gives us the ratio of 9 to 3 to 3 to 1. And this is famously known as the dihybrid F2 ratio, which tells us with such a ratio like this, it implies that we had 2 heterozygous parents which are crossed to give these ratios. And this is often a key feature in solving IB biology higher level genetics questions for unlinked genes. The next video will deal with what's called a test cross. The chance to subscribe to the channel is in the center there. I'm very much appreciated if you do that. And on the right hand side, we have the playlist for all of our genetics videos.